And welcome to session 3.2, AI and Machine Learning in Rare Disease Research. We are so very happy to have all of you with us today and are just so excited to have this conversation. Ahead of the slides coming up, we did just want to give you a heads up that one of our presenters, Annastasiah, is having just a little bit of tech troubles today, so she's going to be calling in and we'll have her slides up, but she apologizes for you not being able to see her live. However, we're really excited about this conversation, and you do get to see our faces as well as Harsha and Swag, our other two wonderful presenters, so you will not be without any sort of visuals. So we're going to give everyone just one more minute to get into the room, and then we'll get things started. Annastasiah, I think we have you as well.
You have me on the phone, and I'm still trying to figure out what's going on. So when everything goes well, I'll probably switch to the computer, but for now you have me on here. I'm really sorry.
Not a problem at all. You know, I think we have come out fairly unscathed in overall tech bumps throughout these 2 days. So, you know, we are all gonna work through it, and just very excited for a great conversation and for having everyone's perspectives here. So just to go over a few brief housekeeping remarks before we get everything kicked off for the rest of the day. So I am Ann Wolf with Global Genes. We also have my colleague Pam here as well, who is our Vice President of Marketing, and we are so happy to have all of you here today, as well as our presenters, Swag, Harsha, and Annastasiah. And so thank you to everyone who is tuned in for this session. We know it's a Friday afternoon. But I am 100% positive that this is going to be a worthwhile conversation. So we thank you for coming. A reminder that we will have everyone on mute, and so questions should be directed via the Q&A or via the chat box. If we are not able to address all questions, some of them we may not know the answer to, or if we run out of time, we will note all other questions for follow-up and do our best to address them moving forward. The structure of the session today, we'll have a couple of presentations, and then we will move into the discussion panel, the discussion portion of the conversation as well. We do have some prep conversation starters, but really encourage you to ask your questions and take advantage of this time to hear from folks, learn from the experts, and get your questions answered. And so without further ado, I am so pleased to introduce our three presenters today. Annastasiah Mhaka, the founder of the Alliance of AI in Healthcare, Harsha Rajasimha of the Organization for Rare Diseases India USA, and Swag of Crayon Bio, who I am going to save from me butchering his last name by passing over it. But so without further ado, we'd just love to welcome everyone, and I believe Annastasiah was scheduled to kick off the introductions, so I will turn it over to you for a little bit more of detail in your background. Annastasiah, are you able to see any of the slides, or is it helpful for us to tell you what slide we're on?
I can't see, but for this one, I would just introduce myself. And Harsha, I'm sorry to impose. You have a draft version. It has some errors in there, but I think that's the best we can do at the moment. So for now, I just wanted to thank you, Global Genes, for gracing us with this opportunity, and to all the research advocates and patient advocates out there, thank you. So I'm Annastasiah, co-founder and former president of the Alliance for AI in Healthcare, as well as founder of Mwambo, which wear several hats at the coalface of emerging technologies and healthcare. So my passion is to impact global health by helping drive lower cost, effective and accessible solutions to prevent, manage, and to cure disease. And let me clearly state for everybody's benefit, I'm not a hands-on expert researcher anymore. Neither am I an expert in rare diseases. But like most of you, I am a huge advocate for the sector. And I'm here today to stand with you and fight with you to identify meaningful solutions for this underserved patient population. So did you want me to go ahead, or did you want the other panelists to introduce themselves?
Sorry, I muted myself. I think let's go ahead with the slides since Harsha has them up. And, you know, again, thank you so much for taking the opportunity to do this, and, you know, for doing this a little bit blind. We'll walk through this together, and appreciate everyone. So Harsha, I'll let you.
Anastasia, I have your first slide ready on the current state of BioPharma R&D.
Okay. Okay, my computer was beeping at me. Okay, so I have to go on memory now. To effectively explore the topic of AI in rare diseases research, we must consider the conventional end-to-end R&D process, including real-world manifestation. So for common grounding, and so we're all on the same page, I wanted to quickly walk us through this traditional R&D framework. It is a complex, long, and expensive process, and it is sequential with no shortcuts, as Swag would tell you later. So elaborating on what you see and depicted in panel A, R&D starts with hypothesis generation by scientists, followed by target identification, molecule design and its optimization, and finally, preclinical studies to assess efficacy and safety in vitro and in animal models pursuant to filing an IND. This takes over 5 years, requiring hundreds of millions of dollars. Panel B depicts the clinical development piece, comprising of 3 phases in humans to ultimately assess safety and efficacy at scale. So these human trials tag on another 5 to 7 years and hundreds of millions of dollars for each new molecular entity. Each of these steps toward FDA approval is risky in and its own right. And the cumulative risk across each of these implies ultimately an overall success rate of less than 10%, taking over 10 years and costing in the range of $2.6 billion. Of central importance is that target hypothesis, which you see at the top of that first slide, is only validated in phase 2 efficacy studies and in real-world evidence generated post-approval, depicted in panel C. So given this long duration, late-stage attrition, and associated costs, we are looking to AI to alleviate R&D burden. So there's a significant amount of effort with over 200 companies developing or applying AI to the R&D process to make it faster and cheaper while delivering more new medicines. Harsha, I don't know where you are. Can you let me know where?
So I'm going to the next slide, the desirable state.
Okay, so, and that slide, I think that's a draft. So I do want to clarify that it's a desirable state, and really for me, I envision a future where the process is no longer linear or sequential as we saw earlier, but puts the patient at the center throughout the AI-enabled R&D process. So with an ability to ingest, cross-reference, and interrogate multiple sources of information into meaningful insights during the development, R&D development phase, we can ultimately predict toxicity and efficacy throughout that process. So information is at the center, and our patient is at the center. And that information gathering can involve things like clinical trials, conference abstracts, EMR claims, lab tests, etc. But ultimately, in this patient-first scenario, the patient diagnosis in the real-world setting sets the stage and becomes the key driver for discovery and development. So this ultimately affords a truly patient-tailored solution. AI and ML can help beyond the initial diagnosis to prognose, help design treatment, follow the patient in the real world, and ultimately yield population-level data and insights. If you look to the right side of that picture and looking at discovery, and I believe that is panel A, and I'm sorry I'm going on memory here. And looking at panel A, you realize that in drug discovery, machine-aided real-world medical, scientific, and business intelligence can then be used to generate much improved and somewhat validated hypotheses to modulate biology and disease. So this might include things like multi-omics, phenotypic screening, polypharmacology. With this ability, this permits us to do drug repurposing with companies like Cyclica and Recursion leading the pack. We can do de novo drug design with companies like Insilico, Blackthorn, very active in the space. And another example is using AI and ML to design and run experiments such as BenchSci and Synthes are doing. So I'll move over to the left side of the slide where you have clinical development. And really the point here is to elaborate that trial design, efficient recruitment, and subsequent monitoring are critical areas for drug development. So recruitment and enrollment using parameters to identify well-characterized patients, measurement and monitoring using biomarker and image endpoints, and notably things like synthetic controls where Unlearn AI is using digital twins platform. And in recruitment and stratification, companies like Deep6 AI are using clinical data and AI to actually target and enroll the right patients. And lastly, remote monitoring adherence, companies like AI Cure. So in this session this afternoon, we will hear from Swag. So this is a fly-in, apparently, Harsha.
Yes, they did.
I don't know if you're showing the whole slide or not.
Yes.
But Swag, in the first part of the discussion, Swag will really talk about mechanistic clarity. This is a vision he has, and he has a definition that he wants to make sure we all adopt as he discusses this. And then the second part of this, Harsha will cover clinical development with focus on logistics. Before I go further, and Harsha, I think you should just get rid of all the fly-ins on this one because it may mess us up because I can't see you. So you can show the whole picture if that's all right,
Yes.
and I'll talk you through it.
It's up there.
So yeah, so before I hand this over, I just wanted to reiterate that we are huge advocates for rare disease, and I challenge us to continue to champion for these patient communities and communicate as effectively as we can to assure that we can shore up global support and resources to push the sector forward. And to you in the audience, you are the leaders in patient advocacy and engagement, and we should really leverage this moving forward. And I think the advantage and the level of communication we need to have is rare diseases are relatable and a huge source of compassion. So I'm looking at the top left of the slide. And with 10% overall prevalence, we are all bound to know someone who is afflicted with rare disease or somebody who is a caregiver for somebody with rare disease. And despite 75% of diseases being attributed to genetic origin, we only have 5% treatment. So there's a lot to be learned there. And Swag will go into more detail on that. And then secondly, rare diseases impact a population that we are all compassionate and care for, children. So 70% are pediatrics onset, and 30% die before the age of 5. So this is something that's absolutely critical to all of us. It touches all of us. It is relatable. More importantly, and this is now on the right-hand side of that slide at the bottom, I wanted to highlight some characteristics of rare diseases for us that make it a good teacher and a source to develop proof points or drive lessons learned. So ultimately, we are applying AI to uncover hidden disease, patient, population, environment correlations, and deriving new actionable insights to ultimately achieve patient-centered R&D while bringing new medicines. As such, all these prevalent diseases will become rare based on attributes and classifiers that we're applying from AI. So in other words, in that first bullet, all diseases will be smaller in size and dispersed geographically. The second piece is around data and standards development. Obviously, we can't talk about AI without alluding to access to high quality, exquisitely curated, privacy preserved, diverse health and consumer data to unlock innovation. And lastly, we need to assure the right tools to solve the right problems, further accounting for things such as transparency, trust, bias, fairness, ethics, and the tools we choose and use in our decision making. So with that, I do want to hand over to Swag with the key observation that we have lots of data and information, especially from advances in omics, synthesis of molecules, and molecular biology techniques. But we still have so few medicines to treat our patients. So why is this the case? And perhaps, Swag, you could share your passion and case for mechanistic clarity to support oligo-based medicines and give us a sense of where we are, where we are headed with a truly relatable case study. Thank you.
Thank you so much. And I have to say, I am truly impressed with how many times you referenced exactly where things were on those slides. So thank you for an amazing blind presentation, and a special thanks to Harsha as well for helping out with the slides. So I thank you so much. Not to hold you back from starting, would love to turn it over to you. You're still on mute.
Okay, excellent. So thanks Global Genes for organizing such a wonderful event. On the virtual setting could be a little bit of a challenge, but I think so far it has been a really remarkable experience. So the question I'm going to pose for myself and the rest of the community is, can we engineer precision medicine for rare diseases fast and cost effectively? And I think I'll convince you in the process that the solution to this problem does not limit to the initial question's realm of application, which is rare diseases. I think, as Anastasia was alluding to, it becomes very quickly a broader solution once you look into diseases in a more gene-centric point of view. So for process that Anastasia already mentioned, it's a long process, and it's a linear or sequential process, and there are several aspects to this. it's something right. There are several aspects of this, from target identification, target validation, and so many steps in the preclinical development of drugs. And then on top of that, there is these phase trials, and it's costly. So I'm going to quickly go through the process, keeping in mind where quantitative methods, machine learning, AI can help understand the complexity of the biological system, treating biology also as an engineering problem, and drug discovery as an engineering problem, where there are design constraints and there are constraints coming from the complexity of the system, just like we are very used to in material science, for example, or physics or electronics. So the target identification, the challenge, the question is, what is a good target? What does it mean for something to be a good target? And there is a lot of input coming in from human genomics data, which obviously is highly enabled by quantitative methods and quantitative readouts. And coming back from the patient data, loss of function mutations in the general population, understanding of systems biology in the human disease context, to early target identification and kind of ranking the quality of target for a particular disease. And the status, I would say, of the field here is that there are lots of genetic targets are being discovered from human genomics and GWAS and stratified population, whole genome studies and exome studies and so on. And quantitative methods here have played a critical role in the effects of variants, and they're getting increasingly well characterized. So it's a promising direction. On the target validation side, the questions one usually asks is the target a good target to modulate, and is it druggable? Is it leading to the outcome desired in the disease state? And there are drug modalities which might have epistatic or pleiotropic effects for the gene target, for example. And the status of the field here is that high throughput phenotypic screening and machine learning methods are enabling mapping of genotype to phenotype, especially in cell assays, and beginning to have a huge impact across industries. So phenotypic screening always has played an enormous role, but now it's enabled, I would say, in automation and readout complexity by traditional arenas where machine learning has been very successful. For example, image analysis of cell assays. And the same story holds for biological assay development in general. So I'm going to run through the slides a bit quickly. If anybody have questions for me, I'll share them because there's a lot of material here. And I just wanted to give a quick overview of where the contribution of these methods are at each step of the process. So assay development itself, obviously NGS technologies have enabled assay development tremendously. And then automation has enabled, as I was mentioning, fluorescent or dye-based cell imaging assays to understand or at least characterize or map phenotypic readouts of a disease state. A lot of time in the pharma world is spent on these three processes, from compound screening to lead optimization and the identification steps. And here, quantitative methods in general has actually a long history. So I don't want to, be this pro hype on machine learning and AI. Quantitative structure activity relationship has been used in the industry for a very long time. However, the recent advances in generative models using deep learning methods and computational design within at least a class of compounds has shown moderate to encouraging success. And predictive toxicology in general, because of a lot of data that has come out and beginning to get shared, machine learning traditionally is very good at understanding, or not understanding, but at least characterizing complexity. So at least retrospective screening data sets has been used to enable predictive toxicology across the industry. And then I think a more interesting direction is also understanding physiochemical properties using these data sets to kind of automate the process of chemical descriptors and features, which previously was expert curated or expert driven. And there are certain areas where this automated feature extraction that deep learning promises has a lot of value because of the complexity of the chemistries, medicinal chemistry in general. I think there is huge strides that are being made in reducing the cost and time of screening. in this process. And also novel applications of existing molecules in a completely different setup. This is a especially illustrative example, halicin as an antibiotic. This was MIT work, which the molecule was known, but the use was novel and it was driven by in silico methods. Validated, obviously, as anything here on biological assays. And I'm going to quickly go through this. There is, of course, a lot of work on ADME, absorption, distribution, metabolism, excretion, and PK and PD of drugs. There's quantitative models, but they're not necessarily predictive models. And in general, in this area, in the preclinical development, later stages of the preclinical development, when you have to have rodents and non-human primates toxicology done, these are expensive experiments. So creating data is expensive. So this is not something machine learning AI community deals with necessarily, taking pictures of cars or streets are cheap. So being data rich in biology can have a real cost, which limits how much one can do with traditional machine learning approaches. So one has to think hard how to do better with less data. So in the clinical stage, obviously, the stages are about assessment of safety and efficacy in humans, and their clinical trial design is getting increasingly, I would say, influenced at least by quantitative methods in general. And I think that some of the promising directions are in creating digital twins from health record data, and I think another area where there has been a slightly longer, I would say, impact is longitudinal high content digital health monitoring to kind of refine and hone on both endpoints of your clinical trials or natural history studies. But the question remains that why do we have so few new medicines from so much of disease understanding? That obviously there's a lot to learn. Human biology is very complex. I'm not saying target space and understanding human genetics is a solved problem. However, there's a huge influx of targets and genes. However, the translation industry-wide is extremely poor, I would contend. It's very slow. So as an illustrative example, these are 4,000 genes, and this is adopted from this recent review, that are associated strongly with rare diseases. However, the number of new drugs, new medicines are lingering in the order of 60s or 70s and industry-wide in a particular year. So why can't we help more patients today? So there is an alternative storyline where, patient comes in, and this is not pie in the sky, obviously, because it's actually happening. And this is Project Baby Bear. Most of you are familiar with this probably know more about it than I do. But an infant comes into the NICU, a rapid whole genome sequencing is done, and I'm going to define this term mechanistic character. There's some understanding of the mechanism that led to the disease. And in 2019, for example, this Project Baby Bear study, and the numbers might be a little bit off because I'm sure it has been updated since, 43% of diagnosis led to what would generally be considered an understanding of the genetic underpinning of the disease, so some understanding of mechanistic clarity. And that took 3 days. I mean, that's an incredible pace, right? Less than $1,000. And still, we don't know exactly when and how long it will take for any of these to be translated in medicine. In the typical timescales of at least more, not ultra rare, but rare and complex disorders, it's 6 to 12 years and hundreds of millions of dollars. So even with mechanistic clarity, that means even not having a problem with understanding the cause of the disease, there is this lack of translation speed and cost-effective solutions to it. So just to be on the same page, what I mean by mechanistic clarity is it's pretty simple. It's just rare disease etiology is understood by the effect of the mutation on genetic function, a gene-centric view, which says the disease biology is because of XYZ, a loss of function or haploinsufficiency, gain of toxic function, dominant negative mutation, and basically leading to the lack of a functionality of a particular molecular biology machine. And another way of looking at it is by the impact of these mutations on the phenotype of interest. So here, this is a very nice review of kind of this way of looking at the problem, which is a gene-centric approach, which says, well, there are a bunch of mutations, there is gain of function mutations, a loss of function mutations, and that leads to a diversity of phenotypic effects which can be measured and cell lines in human population by biomarkers and so on. The measurements themselves necessarily sometimes are challenging, but not necessarily the key challenge. But having this understanding of genotype to phenotype, this mapping, is not always obvious, but we are getting increasingly better at it. And then genotypic feature model tells which gene is a good target and prioritize targets and so on. So this gene-centric view does allow us to treat biology as a more engineering problem, be more complexity driven, which is how the rest of, let's say, any other engineering process to make something new works, including your car, including your laptop. And this complexity-driven understanding can hugely impact drug discovery, because the idea is that there is a lot of data that can inform the output as long as it's a good quality data, but there is a process which is not simply one gene, one effect. There is a more complex outcome. And this understanding of obviously is also translating and revolutionizing complex disorders in general. And here I'm showing from the same paper, this review of genetic disease, it's a really nice review, where we have 80,000 associated variants of significance today for complex diseases. And then these are getting increasingly mapped into phenotypic understanding of either altered interactions between genes and proteins, loss of function of the loss of interaction or loss of function of the protein, gain of interaction, or regulatory interactions that are getting modulated. So this is obviously a very involved field of research that's still evolving, but I would argue that we are getting increasing clarity in complex disorder along these same lines of mechanistic clarity. Now the question then becomes, well, if we have so much clarity, are there modalities, are there drug modalities that can precisely, immediately, and directly modulate gene expression? And there is, oligonucleotide-based medicines, this are siRNAs, ASOs, aptamers, and so on, can actually directly affect gene regulation. And there are many mechanisms of this. I want to go through the details of it, but it's well established, Watson-Crick hybridization. The mechanism of the drug action is well understood, I would argue. And there are several mechanisms within these effects that are either occupancy only, which leads to splice modulation, or enzymatic action, which needs to knock down in the case of siRNA or ASO. Or splicing modulation, which is what I'm showing by this animation. So it works on, the key point, it works on the mRNA, it works on the pre-mRNA, right, which traditionally is undruggable. It works on non-coding RNA. It can be highly allele selective. So we're correcting for toxic gain of function diseases. It's doable. It has generally good distribution, at least antisense oligonucleotides in kidney, liver, eye, brain, lungs. So, and it can be used to additionally engage other enzymatic systems. And the story is, it can be personalized for a patient. All of you probably know this story of Mila, when she had a rare disease of Batten's disease. It was a homozygous mutation, but heterozygous in the kind of mutation. She had a retrotransposon insertion that led to early termination of an essential gene, CLIN7. In her case, that led to Batten's disease phenotype, but the genotype is unique to her. And Tim Yu's work, it's fabulous that this led to, within a year or one and a half years time scale, a customized antisense oligonucleotide that could indeed correct the splicing defect that she has, that's specific to her. So this is a mature field, however, right? So this story is built upon a rich field that several companies have worked on for a long time, Ionis, Alnylam, Roche, and so on. But it really started in the early 80s, where DNA oligonucleotides and RNA-seq activity was understood, or at least first reported, and then increasing understanding has been gained over the last 3 to 4 decades. And here's a short timeline, and these are medicines that have been researched for decades. There is a rich understanding of mechanism. They're clinically proven. So what is stopping then this this modality to be really being scaled up in the way that I just showed for one patient. Well, the problem, of course, is that as with anything in medicine, that these are most of the time not active, and depending on the modality or depending on the particular chemistry, I'm showing here a particular example because the of course numbers will vary, they are mostly toxic, right? And as I said, the toxicity is different for different chemistries of these modalities. But it's a complicated business to create what seemed like a simple job to create millicent, for example. It's not that simple just because there is a lot of tox endpoints, cytotoxicity, membrane toxicity, immunogenicity, and immunostimulatory effects, and then several efficacy contributors and things that one needs to optimize. So if you think of it as an engineering problem, there are several design criteria and engineering optimizations that needs to be done for a drug of these modality, nucleic acid drugs, even to be considered a good design, a good drug. And in the efficacy, there is, of course, tissue distribution and production and productive uptake of the drug, targetability of the RNA. Is the target site a good site? And then hybridization, specificity, enzymatic rules of what would the enzyme still cleave? When will the drug's mechanism of action be on target and off target? Basically, it's a specificity question. So that comes to what we are doing. So conflict of interest disclosure. I am the co-founder and chief scientific officer of Crayon Bio. This is a very early stage drug discovery company. And our goal is to make precision medicines on demand. And the focus right now for us is precision medicines in the space of oligonucleotide-based medicines for reasons I just enumerated. I want to recognize this is built on the shoulder of giants, that with any work in science, several companies have worked in the space for therapeutics and have made a lot of progress. And we have a unique vision, I believe, right? That we want to engineer eventually new personalized medicine immediately following diagnosis. That's our goal, that's our vision. And our mission is to make such gene-centric medicines affordable. That's very important to us, and rapidly available to patients globally. Globally is also very important to us. We don't want to make these rare disease drugs for only a certain population of patients in the world, or a healthcare system that can sustain an exorbitant cost for developing such medicines. So we are trying to create customized high throughput assays and data sets, and we have created algorithms. We have created methods for mapping and then thereby generating predictive models for OBM pharmacology. So the analogy here is very data driven. If you wanted to build a self-driving car, you would drive it through streets, show it enough stop signs, hopefully show it enough schools, and eventually, and pedestrians, and eventually it might have enough data to learn the rules. Obviously, here the environment is changing, so the analogy breaks down. Here, at least, the levers you're pulling has persistence in time and design, which means the levers you're pulling here are the different chemistries, the medicinal chemistries one can use for tweaking the pharmacology of oligonucleotides-based medicines. And these are backbone modifications, base modifications, sugar modifications, and so on. And there's so many of them, and there's so many possibilities. The main challenge is the design space is huge. Again, analogous to creating a self-driving car, where the constraints in the design space and safety concerns are somewhat, I would say, at least analogous. That you have to make something absolutely safe for it to be of any interest. So imagine a world where you could do this. It just, obviously this is still not, well, it's not entirely not possible, as you saw the Millicent story, but imagine a world where a patient could come in and you get mechanistic clarity from genomics, systems biology, and other methods. And you know what genes to modulate. And imagine you could just, and this is a rapidly dropping cost of doing this today. Imagine a world where it takes days and not years to just print out a drug that, and we can get into the details of that, which we don't have time in this session, obviously, of how that would be a quote unquote a pre-approved modality of composing units that independently are known to be safe, where it reduces to basically good manufacturing practices, manufacturing cost of all such nucleic acid drugs. obviously the impact of that is huge. There is so many diseases, as Anastasia already mentioned, and we know a lot of these are genetic in origin. I mean, a lot of these genetic understanding is still an evolving field of study, and we're getting more clarity on it, but it is strongly genetic. And there's a strong prevalence across millions globally. However, very few do have treatments today, right? And it disproportionately affects kids. And this paper, this set of papers, both of them, this one especially, basically shows there's already lack of target is not necessarily a problem. There's 6,000 protein associated at least with one rare disease. There's a long distribution of incidence rates of this disease. So there's a long tail, which you integrate leads to this idea that there is a 1 in 10 kind of rare disease, right? So with that thought, I'll end. I wouldn't have time to dive into the details, but happily the slides, my email is swag@crayonbio.com. So please reach out if there's interest. I know the session and the presentation format is a little bit limiting for online discussions, but I'll happily take questions later.
Thank you so much. So happy that Anastasia has also made it to our camera for anyone that did not see that. So, but in the interest of time, we are at about 15 minutes out. So Harsha, I'm going to turn it over to you, and then we'll get in a couple of quick questions at the end as well.
Sounds good, and thank you, Anastasia and Swag, for covering bulk of the process of bringing new drugs and the inefficiencies and opportunities that have emerged recently with oligonucleotide-based medicines and others. Certainly, it was very powerful to hear Mila's story from her mother in the opening session of RDDS as well. So what I'm going to do is in maybe 10 minutes or so, I'll make my comments and leave some time for questions at least, so we can hear from our audience. My talk will be on artificial intelligence in rare disease research powered by patients. So some of you may have known this. I started as a genomics data scientist and grew up in analyzing genome sequencing data. I'm primarily a software engineer turned into genomics data scientist, and I was going about my day-to-day life as a scientist at National Institutes of Health. In 2012, I experienced the loss of a child to a rare congenital disease, and that got me into the rare disease advocacy side. So since then, I set out on a mission to apply my years of postdoctoral training towards accelerating clinical development timelines for both therapies and diagnostics. And over the last 7-8 years, I have been flipping this question of where AI or technology is useful to where patients have the biggest need. And so the innovation bubble that we are in right now is there is a booming innovation industry, a new startup created every day, a new app being launched every minute, but the actual experience of delivering or receiving care is changing scarcely. And so that's what really drives me. And we need to ask the question of when do we really need artificial intelligence? And more importantly, when do we not need artificial intelligence? You know, I remember around sometime in high school when we switched from doing math by pen and paper to calculators. And so there is a limit to what the human brain can do. And just because something is being done by the computer or a calculator does not mean that it is artificial intelligence, but it could be. I guess the definition broadly, AI has become a more public domain term now. And if that's some people do define AI in that manner. And certainly there is huge opportunity to apply computational methods to speed up certain calculations in this process. So what I have been doing over the last few years is looking at why rare disease drug development takes much longer compared to non-rare disease drugs. And when we started reaching out to hundreds of customer stakeholders, we heard quotes like, every trial feels like the first ever trial undertaken by mankind. And recruitment is the biggest problem every single time in a clinical study. and a patient advocacy group saying we have all the tools, yet the burden to participate in clinical trials remain very high. So now, where does artificial intelligence really come in to address these problems? If you start peeling the layer of the onion, you see that travel is the big problem, and about a quarter of the patients who review the informed consents are unable to participate because of the number of site visits or the location of the site. And so to solve this, we are developing methods whereby we can move the needle more towards the patient and where the patient's convenience is, wherever there is an opportunity to not require a in-person brick and mortar site visit can be taken to the patients on the smartphone or the mobile device apps. And eventually delivering the investigational medicinal product or sample collection and so forth could also, all of this is kind of being done right now in many of the clinical trials, as you may be familiar with, all in sort of a la carte piecemeal approaches, but it's being done in a manner which is not necessarily reducing the burden on patients. And so that brings us to the patient-focused drug development paradigm. If you look at the overview that Swag and Anastasia presented, there is a ton of companies, and this is not a comprehensive list by any means, number of innovative startups and bigger companies working at various phases of the drug development and discovery process, leveraging both the scientific literature, the scientific data that's published and available. Much of it is not, particularly when you get down to patient data and health outcomes data. Those types of data are very hard to gain access to, which are often the need for training your machine learning AI models. And so having good data is super critical. And in the case of clinical trial timelines, why they take so long, if you look at the patient recruitment, retention, and compliance, the attrition rates where patients drop out, that has a huge impact on the ability to complete a clinical trial successfully. And so when you see the failure rates of about 90% of drugs not making it to the FDA approval, that these attritions have a big part of that, and why it takes so long. And that around the comment of every trial feels like the first ever trial undertaken by mankind. Of course, we are not learning from past clinical trials, and we are not continuously improving the process of bringing these drugs. It has largely not been industrialized, ironically, although there is a huge industry around it. And so that's what we are looking to do, is gather the operational data as much as the patient level data to understand where patients are dropping out of this cycle and how those dropouts and delays can be addressed. And this is where AI can really come in handy, in my opinion, as the human brain's ability. And there is no one person who is responsible for this. The process of bringing a drug to market is probably the most complex endeavor undertaken by mankind, and hence it's kind of sitting in the brains of multiple people in every single trial, which makes it a good candidate to automate and use AI ML methods for that continuous improvement. So I'm taking a quote from Vaz here, but this is largely accepted by most people in the industry, is that the first thing is the importance of having outstanding data, and how people underestimate how little clean data there is out there. We hear about all this big data and machine learning and AI. There is so much of hype around it, but when you start looking at how many patients do we have for whom we have all the genomic sequencing data and the behavioral data and their social determinants of health and the environmental factors, that will bring down the number of patients' data to a very small number. And how many of that data set is actually gathered in a way that's usable, that we can now query and build our AI models. And so that's something I think Dr. Chris Austin also emphasized in the opening keynote around the use of patient registry and natural history databases to creating good quality data there would really start addressing a lot of these pain points here. So I'll quickly move through this last solution slide where the traditional way of having patients visiting the brick and mortar site, as you all can probably imagine right now, it's all virtual, and clinical trials are no exception. But to do that, it's a paradigm shift, as almost all of the clinical trial site visits have been in person for patients visiting to their clinics. That's kind of changing to some degree, not a whole lot quite yet, and hopefully the changes around the corner with a lot of the regulatory barriers around telehealth across state borders and other barriers being removed by the FDA to go all the way from pre-screening subjects for eligibility to a clinical study to informed consenting from where the patients are without necessarily requiring a visit, and onboarding them and collecting all the patient-reported data, video conferencing based visits instead of live in-person interactions to do the clinical assessments. And doing all of this in a HIPAA and FDA compliant manner is where there is still a lack of good validated tools and options that can cover all the types of clinical trials that are out there. It can't be a one size fit all, a solution that works for a small molecule traditional biopharma clinical trial and a cell and gene therapy trial and a NF1 type trial. And so we are focused on developing a scalable, flexible solution on the cloud that can enable this type of solution that can now aggregate and track data from past clinical trials that can learn continuously and make the next trial more efficient. So AI is not going to replace drug developers, but drug developers who use AI will soon replace drug developers who don't. And so key takeaways for me is we have come a full circle with FDA driving the patient-focused drug development paradigm. And so we need to keep in mind that technology or AI is a means to an end. And the only way to end data silos is to work closely with the scientists and researchers to ensure data is generated in a complete and quality conscious manner and not a afterthought, but more of a forethought. And orphan drug development is twice slower. Global silos have to be bridged to accelerate clinical development in rare disease. Geographic and mobility challenges can be addressed using digital health and not so much of AI, although you can bring Alexa and other voice recognition AI-based methods or natural language processing methods. So there is a lot of opportunities where AI can be applied in practical ways, but with cautiously choosing and selecting the technology with the patient-focused drug development paradigm at the center. With that, I'll pose a question to the panel and to the audience. Do you think it's AI in clinical research powered by patients or patient-focused drug development powered by AI? Thank you.
Thank you. So I would, we only have a couple of minutes left, unfortunately, but appreciate everyone still here. So, in the interest of time, I would love to just invite everyone to maybe address that, each of you to quickly address that question, as well as just to think about if there's one thing that you really wanted to share with the audience in terms, thinking from the perspective, especially of researchers and patient advocates, who make up so much of our audience here. What is one thing that you would share with them as a tidbit of advice or a place to get started? Because I think this can be, it's such an exciting conversation, but it can also be really daunting if you're not as embedded in the data as I know all three of you are. And Anastasia, I'd love to welcome you to maybe answer the question first now that we have you on camera and can see your face.
There we go. Yeah, so thank you. So first of all, sorry for all those difficulties. So it's good to be on here, and I hope I didn't throw everybody off. I think the key issue, and I just want to reiterate, and I don't know if everybody heard this clearly, Harsha said, do not have the data issue be an afterthought. It should be a forethought. So as you design your experiments, as you start doing your research, you have to prepare early. And there's a lot of thought that needs to go into that, because when we annotate data and we sort of collect it with what we know today, we're actually limiting ourselves a little bit, because the whole idea is AI can uncover and unlock patterns that we don't anticipate or see. So as we're collecting data, we do this. I'll give a very crazy example. There's a COVID consortium for critical care. They have 305 data elements, and the physicians have to enter 110 data points every day. They're following everything. And where they're really leveraging AI is AWS and IBM are helping with the data and the natural language processing so that the doctors can quickly get the data in. And they have such a treasure trove that they're following these patients. They're going to follow them out for several years, look at all their late stage organ issues that arise. So the idea is, I'm not saying that it should be that many data points, but when you can exquisitely annotate and curate data, you're already a step ahead, because if the quality is not there, it's just not there. So I'm not saying anything that anybody doesn't know, but I think that's critical. And then in terms of AI, I think there's also a huge, we've had a huge hype. And part of our job is to sort of control and manage that hype. And so for me, while I am a huge advocate for AI, I truly see it as a tool, and our research and patient advocates should also view it as such. So answer the right question with the right answer. Sorry, ask the problem question first and then find the right tools for it. So AI is just one piece of a longer story, and sometimes there are tools that are better suited. And then lastly, when you do use AI, be aware of limitations such as the bias issue, the fairness, and the training data set. So those are sort of the key takeaways that I would sort of like to enunciate. So number one, plan up front on your data. Number two, be very, very deliberate about how you annotate and follow it. And then the third piece is that AI is not the answer to everything. Find the right tools. And collaborate. That's the last one. Make sure you work together. Yep, mute Anne.
Thank you. Thank you. One day we'll all remember to unmute yourselves. Today's not that day. But Swag and Harsha, just you were able to just maybe share a brief remark super quickly. We do have to close, but I hate to cut off any kind of last points you want to make.
Swag, go ahead.
Oh, I have nothing to add. I'm good.
All right.
Swag is helping drive shortening that path from 10 to 15 years to days, 2 weeks. I think that's his vision. And I'm excited about it.
Yeah, if we get there, I'll consider my life worth living. So,
Harsha, anything else that you wanted to add?
No, I think I made my comments on what I thought. It would have been great to have audience interaction, but that I guess we ran out of time. So,
you know, unfortunately, we did run out of time. However, you know, just a huge thank you to all three of our panels today for their wonderful insights, for persevering during tech challenges. And, you know, we'll just have to do this again. We'll just have to do 2 hours, leave lots of room for Q&A. I know there's plenty to be said. Thank you all so much. Please, everyone, head back for a closing session. You can access that by hopping back into the auditorium in the platform, clicking the join button next to the closing session. And we have some really great speakers. So please take advantage of that opportunity. Thank you again to everyone as for being here as an attendee and as a presenter. And to the question that was just posed in the chat box, we will be saving that. So we will take the opportunity to make sure to follow up with the speakers on that afterwards and do our best to get you an answer as well. So thank you all so much, and we'll see you soon in the closing session. Bye-bye.
Hey, thank you.